So after shapes, size, number of cells in an organism, one very important structure now I'm going to start with is the prokaryotic cells. As we gave the example of prokaryotic cell is the bacteria. So here, as you can see in this diagram, I am going to share with you the structure of prokaryotic cells. So please pay your concentration in understanding this structure. I'm going to first start with the outer layers. As you can see, this layer, which I have marked with the lining portion, that is known as cell wall. So first structure which is present outer, or we call it as the outermost layer in a prokaryotic cells. Again, I'm repeating, we are discussing the prokaryotic cell. So outermost layer that is present in a prokaryotic cell is cell wall, which is made up of peptidoglycan. As you come inner side, there is another protective membrane which is known as the plasma membrane. As plasma membrane has protective function, it is protective in nature. All these have protective and transportation function. So first outermost layer is cell wall, which is made up of peptidoglycans, and plasma membrane, it is protective in function. There is sometimes in few bacteria there is additional structure or layer present in, which is slimy layer. If it is loose, as you can see, this fibrous dot like structure I have drawn after even the cell wall, that is called capsule. This is called capsule, which is also known by the name glycocalyx. As you start reading your NCERT, it will be very much clear to you what I am talking about. That outermost layer, which is present in few bacteria, it may be present or it may not be present. So if this is thick and tough, we call it capsule or glycocalyx. If it is a loose sheath, then it is known as the slime layer. It is an additional layer. Again, I will repeat, this capsule or glycocalyx, it is the additional layer in the bacteria. So I hope that three layers are very much clear to you. Now let's move to the inner side. The structure, or the basically transparent material present throughout the cell, that is called cytoplasm. As we said that prokaryotes, it does not have any organelle. So all the enzymes for different activities, they are present only and only in cytoplasm. So cytoplasm has many enzymes for various metabolic activities. As further you can see, this layer is plasma membrane, and this has some foldings, inner side foldings. So at few places this plasma membrane is folded, and these structures, they are known as mesosomes, or I will say these infoldings are known as mesosomes. Three function quickly for mesosomes. They have enzymes for replication. They work like mitochondria because they do not have mitochondria. So here mesosome, it works like mitochondria. It helps in it helps in cell division. Why it helps in cell division? It has DNA, circular coil DNA attached with it, and DNA is deoxy ribose nucleic acid. So this DNA, it help in replication. So mesosome, it has enzymes for replication. It work like mitochondria and it helps in cell division. Then because not having membrane bound organelles, so here in the cytoplasm there are few structure, and this structure is called ribosomes. And ribosomes in prokaryote is 70S type. 70S. You will ask, ma'am, what is S? S is called Swedberg's coefficient, or it is also known as the sedimentation coefficient. Sedimentation means at which density we can uh, contain it, at which density it has two parts. So that I will take further as we'll move with further with the chapter. So for this right now you need to only know that ribosomes are present in prokaryotes and they are of 70S type. Then it has another extra circular DNA which is known as plasmid. It is called the plasmid, another circular extra nuclear DNA. So this is all about the structure of a prokaryotic cell. Then there are a few terms which you need to remember regarding this. If the plasmid is present in a bacteria, those cells are positive strain, and if plasmid is absent, those are negative strains. There are different kinds of strains. Suppose if I have to say there is a one microorganism which can grow on this medium with the which is having this thing. If plasmid is present, that will be positive. If plasmid is absent, this will be negative. Then there was another scientist called Graham. He gave us that we can stain a prokaryotic bacteria. So when you stain a prokaryotic bacteria, those which take up that Gram stain, they will be known as Gram positive bacteria. Those which cannot take up stain, those will be known as the Gram negative bacteria. So bacteria can be of two types, Gram positive and Gram negative. So that was about the prokaryotic cell. As I'm not going to start today with the eukaryotic cell, so here there is few homework which I will provide you. So these notes you have to write down as your classwork with the diagrams you have to always and always draw with pencil, and this will be mentioned as the classwork, and homework I will provide you in the next sheet.